There we go. Okay, so we've just done meiosis 1. We've just finished making from one parent cell, and we just yielded 2 daughter cells. Okay, but each of those daughter cells still has, all right, excuse me, there we go. Each of those daughter cells still has 2, I said 2, 23 chromatid chromosomes, all right? Basically meaning, all right, here's a chromatid, that's 23 chromosomes, here's the other chromatid, all right? One of those is going to be replicated, okay? So that's what we're still, even though I know the number up on the slide can be confusing, all right, each is 23 replicated chromosomes with 2 connected sister chromatids. Because remember, we replicated our DNA. So that means we have the original plus we have the replicated. Okay, so the original is 23, replicated is 23. Okay, still with 46. So now we are going to, this should not say mitotic, that should say meiotic. All right, now what we need to do is we need to separate this structure. Okay, we're going to separate the chromatids and finish up with our haploid cells, meaning each cell in total has just 23 chromosomes instead of where we're dealing with 46. Okay, we'll go into single chromosomes then. All right, so let's start off. PMAT, all right, prophase 2, all right, similar to what we saw in prophase 1. All right, again, not mitosis, that should say meiosis. All right, we're going to break down the nuclear envelope because that's where our chromosomes are stored. All right, we are going to collect our chromosomes and they're going to start to group again. All right, but now in this scenario, this is very important. We've already done the crossing over. We don't need to do it again. Okay, so no crossing over. All right, this is a great test question. All right, don't be tricked. All right, prophase 1. Crossing over. Prophase 2, no crossing over. It's already been done.
It does say not rigid. It does say not rigid. It says the second prophase of
mitosis? Oh, okay, okay. Well, I mean, it also resembles the prophase phase of meiosis, except that there isn't any crossing over that's going to occur. All right. So we don't have to worry about that because there's no more homologous chromosomes. They aren't present. We got rid of those way back in anaphase 1. All right, when we lined up the chromosomes on either side of the cell of the equator of the cell there. Okay. All right, and we started to pull them away from each other. All right. So let me show you a picture. All right. So notice. This is just now we're seeing what's happening in each individual daughter cell. So we have 2 cells. These are both the same. Okay. So in prophase 2 here, all right, we're again breaking down the nuclear envelopes in each cell. All right, and then we now start to see this grouping of these chromosomes here. We're starting to get our centromeres. They are not centromeres, centrioles. Excuse me, are migrating to the opposite ends. They're going to start heading in that direction. Okay. So we're breaking down. We're not having any crossing over in this situation. Now, when we get to metaphase 2, all right, we're going to start to get our chromosomes again. They're going to line up on the equator of the cell. All right, and in this situation, all right, the equator of the cell is going to run through the centromeres, which is that little structure, all right, that it holds each sister chromatid to one another. Okay, so basically you're going to see, all right, those chromosomes be directly in the center with each chromatid one on one side and one on the other here. All right, so very similar like we saw before. All right, the centrioles are going to send out the microtubule spindles there. They're going to attach onto the centromeres of each chromatid there. Okay, and we'll see in anaphase 2. We're going to start to pull, those mitotic spindles are going to pull, all right, and it's going to split the sister chromatids apart from one another. So we'll end up with a single stranded chromosome. So now on one side of the cell you'll have one sister chromatid, and then on the other part of the cell, you'll have the other sister chromatid. Okay? And they'll get pulled to the opposite poles of the cell. Okay? So each one of these contains 23 chromosomes. 23. All right, so that's what we're seeing here. All right, metaphase 2, they line up. The mitotic spindles, again, are attaching to the center of the cell and onto the centromeres there. All right, and now in anaphase 2, we start to pull the sister chromatids apart from one another. All right, we start to form that cleavage there. Okay, as we're pulling the chromatids to the opposite ends there, which brings us to final phase, telophase 2, all right, where we then now take all the genetic information, package it back up into the nuclear membrane there, all right, and then we start to divide the cells from one another, and we wind up with our 4 haploid daughter cells, okay? In total, we have 23 chromosomes. One is our sex chromosome. It's either going to be X or Y, okay? And the other one, all right, is going to be our autosomes there, okay? So in women, all right, this is what we're going to see, all right, when we as a secondary oocyte. And then in males, this is what we're going to see as our mature sperm. Okay, that's what we got. So when we talk about, all right, what's going on, we go through the ovarian cycle here, and you hear me mention certain phases of meiosis, then you'll know, okay, this has already happened at this phase here. Okay, so what happens when things don't go as planned, all right? When we get a failure of separation of the homologous chromosomes, or when we're dealing with our sister chromatids. All right, so we're either going to have an issue, all right, with meiosis 1 or meiosis 2 there, all right? Do we not separate the homologous chromosomes, all right, during division, or the sister chromatids, which we just saw here in meiosis 2, which means we'll either end up with a gametes that's going to have one less chromosome or one more, 22 or 24, okay? So in certain situations, if we have too many, 47 chromosomes, trisomy. We'll talk about the most popular trisomy here in a moment. All right, so that basically says we have 3 copies of one chromosome instead of 2 copies. All right, or we can have 45 chromosomes, all right, which we call monosomy, and that means we just have 1 copy. All right, and all because when we were pulling our cells apart, one was stubborn, and it didn't get pulled apart properly. Okay, it could either be during meiosis 1, all right, where we get the homologous chromosome separation, or during meiosis 2, where we get the sister chromatids to separate from one another. Okay, so the most common one that we see is Down syndrome, trisomy 21. Okay, most of these, and if you go into, I think they might teach it to you in the nursing program. They'll talk to you about the trisomies and the monosomies, but most of those die in utero. Okay, so the fetus will make, or I should say the fetus, the embryo will never develop, and they just go the way of being expelled. All right, but in certain situations, like with trisomy 21, also known as Down syndrome, all right, we'll see these folks here, all right, with several distinguishing characteristics. Usually they'll have some sort of intellectual disability, okay? They'll have what they call frontal bossing of the forehead, a larger forehead. Most of these folks will be quite short, all right? Big issues with the heart. Lots of heart defects. In fact, that's usually for these folks that are born and whatnot and live through their adolescent years. It's not very common to see folks that have Down syndrome, trisomy 21, live into elderly years. And if they do, that's rare, all right? Because most of the time, they have heart issues. All right. Usually they'll have heart valve problems. Okay, or they can have malformation of the aortic arch. Usually those are the things that are caused these folks some issues here. All right. You see it more commonly with the non-disjunction in the eggs and the oocytes, okay, than you do in the sperm. And that's why it is very, very important, especially with moms, all right, that they discourage women from having babies or try to get pregnant after the age of 35. It goes from it's like 1 in 10,000 of having a non-disjunction or a Down syndrome baby to 1 in 100 after the age of 35. Okay, and I know this because my cousin went through it. She was worried about that, so she's a nurse, so she was weighing and her baby didn't, but she was still concerned about it. Oh, look at this slide. This looks familiar. All right, here comes the quick review, and then let me just run through this, and then we'll get into our mock exam. All right. So let's go back. We're going to start with the ovaries first and talk about the ovarian cycle. And this is one of the reasons why I love teaching this to you ladies, because you already know most of this because you experience this. It's men, we just don't get it. We're not smart. All right. No offense to any of the men at home. All right. But it's just different. It's easier to understand it when you experience it. Okay. So let's go through the different types of follicles here. All right. Obviously, all right, the major reproductive organ here are going to be the ovaries. All right. We're going to start off with our primordial follicles. All right. Now, the primordial follicles are going to be, all right, prior to meiosis, they will have, all right, 46 chromosomes here. Okay? So the primordial follicle is going to have your primary oocytes. And it's real simple. It's just one single cell, the oocyte itself, all right, lined by a flattened layer of follicular cells. Easy. Okay? You need to understand that the primary oocyte in a primordial follicle, all right, has stopped in prophase 1. Okay? So prophase 1, if you recall, all right, we've replicated our DNA. Okay? So we've made the copies. All right? We're going to have crossing over and all that, but that's it. It stops right there. Nothing else goes on. All right? So when you're born, 1.5 million. That's a lot. Obviously, you're not going to use all those, all right? But 1.5 million. All right. So the primordial follicle will then develop into a primary follicle, okay? And that primary oocyte, all right, now that flattened layer of cells that surround it now take more of a cuboidal shape, and we give them a name, granulosa cells, okay? And it's these primary follicles that are going to secrete estrogen. That's great. We've got to get the uterus ready. All right, low levels of estrogen. And we're going to talk about how estrogen levels and all these other hormones are going to affect the hypothalamus and the anterior pituitary and all the other developments. We'll get into that, not tonight. Okay? So our primary follicle is going to start to ready, all right, the uterus. Okay? And we'll see just outside of the egg itself, you've got this glycoprotein layer, all right, called the zona pellucida. All right, so you have the oocyte, then you've got this layer surrounding it, that's the zona pellucida, and then outside that, you've got your cuboidal layer of granulosa cells. Okay? All right, so that's what we're seeing here. Okay, here are the primordial follicles. All right, flattened cells that sit outside, and then you've got your primary oocyte. Now, in the primary follicle, you've got your primary oocyte, then you've got this kind of translucent glycoprotein layer that sits right outside, and then immediately outside of that is the granulosa cells here. Okay, this will all be familiar to you. All right, so now we're going to see our secondary follicle still containing, all right, the primary oocyte. But now we go from one single layer of granulosa cells to multiple layers. Okay? The outermost layer, this is important, is the thecal cell layer. All right? They are going to secrete androgens. Remember, androgens are going to be the male sex hormones. Well, that's not good. No problem. All right? We got our granulosa cells that are going to convert it into estrogen. Okay? So not to worry. We're good there. Okay? So we have our female hormones being produced. Okay? Because again, we're still continuing to get the uterus ready, all right, for implantation. So here you can see your secondary follicle. Here's the primary oocyte still inside, surrounded by, all right, the zona pellucida, our glycoprotein layer. Now we've got multiple layers of granulosa cells. All right, the fecal cells outside here, all right, are producing the androgens, but not to fear, the granulosa cells are converting those androgens into estrogen. Okay? All right, now we're going to start to see a space develop inside of our follicle. As our follicle starts to continue to mature, all right, once we start to see a space develop, all right, filled with fluid, of course, we call it an antral follicle, and that space is going to be the antrum, okay? And so eventually, as that follicle continues to grow and mature, eventually the oocyte will be pushed off to the periphery, okay? So when we see the oocyte there, all right, when it starts to get pushed off, it's got a cluster of cells around it called the cumulus oophorus. I think of cumulus nimbus clouds for whatever reason. And so I think of this structure, all right, just being kind of a big puffy structure on the inside here, all right? But we'll see, all right, immediately the cell layer that surrounds the zona pellucida. So it goes primary oocyte, then the zona pellucida, that glycoprotein layer, and then immediately the one cell layer that surrounds the zona pellucida, we call that the corona radiata. Okay? And then all these other cells that are outside of that, all right, is going to be what we call the cumulus oophorus. Let's see if we got a picture. Here we do. Of course we do. All right, so there's the primary oocyte, there's the zona pellucida, then you got this one cell layer on the outside. All right, that's the corona radiata. All right, and then we still have all of our granulosa cells, and we have our thecal cells on the outside. But you can see we now start to form this space here. All right, filling with fluid. Eventually, as that space continues to fill, as this follicle grows and matures even more, it'll start to push, all right, the oocyte off to the periphery here. Okay, and that's when we're going to start to get our mature follicle. Okay, now at this point, all right, once we see that the oocyte has been pushed off to the side here, all right, now we're going to see that the oocyte is going to be labeled a secondary oocyte, and it is just finished, this is important to know, meiosis 1. All right, and I'm going to show you specifically, well, give me 1 second here. I love this picture. If you haven't looked at this yet, you probably haven't. All right, figure 28.6 is wonderful. All right, because, and I know you can't see this, all right, it's telling you, you can see in the order of events that's occurring, all right, with the actual oocytes and with the follicles, okay? And there's a nice description on each side here that tells you what's going on with the oocyte, what phase of meiosis it's in, all right, and what's happening with the follicles, all right? So here, when we just complete our metaphase 1, we're going to see here now we've got 2 cell structures here, right? One's called the polar body. That's garbage, right? There's nothing in there of any use to us, right? Except that there's 23 chromosomes in there because you have our primary oocyte that finishes meiosis, and we just finished talking about meiosis where you started with 1 cell and then you wind up with 2 cells. And so that's what we see here, all right? Our primary oocyte has now delivered 2 cells. One is the secondary oocyte that has 23 chromosomes in it, and then the other one is the polar body, okay, which has 23 chromosomes in it. But this will not amount to anything, okay? Has anyone ever seen the movie Twins with Arnold Schwarzenegger and Danny DeVito? All right. Well, I was going to say, this is Arnold Schwarzenegger and this is Danny DeVito, all right? But anyways, since you haven't seen it, I have to stop there. Can't go on further. Where are we? Mature follicle. Okay. So you notice here, okay, that, whoops. All right, our oocyte is pushed off to the periphery here. All right, we're done with meiosis 1. We start to go into meiosis 2, and we stop, all right, in metaphase 2. And you're going to stay there until menstruation. Okay, so every single oocyte, all right, was going to be arrested in metaphase 2. All right, and that's as far as it goes until, all right, if this follicle is lucky enough to be chosen, okay, it will remain in metaphase 2 until menstruation. Okay, and so that you're only going to see one of these occur a month, well, during the period. All right, and then once we have a mature follicle, and that mature follicle eventually comes to maturation and it ruptures and expels the secondary oocyte, what's left behind is going to be our corpus luteum. And believe it or not, the corpus luteum plays a role of an endocrine gland for a short period of time. I believe it's up to 3 months. All right, we'll talk about that in a little bit. All right, but after the mature follicle ruptures and the oocyte is expelled, it's this structure that plays the role of the endocrine gland. All right, it produces progesterone and estrogen. All right. Again, this will help with that uterine lining here. All right, especially now we're very close to fertilization. Okay, because once that oocyte is expelled and it enters into hopefully the fimbria, all right, the infundibulum and the ampulla and whatnot, all right, which is where we're going to see fertilization take place. That is what's going to occur. All right, we only have a couple of days to have that occur while the uterine lining is prepared, ready for that to happen. Okay. And then finally, if no implantation occurs, all right, then the corpus luteum degenerates into what we call the corpus albicans, which is basically scar tissue here. And in most cases, all right, all that corpus albicans is resorbed, goes away. All right, so this is all review, all review. All right, so when we discuss, all right, meiosis, especially with gamete reproduction in females, we call that oogenesis. Okay, where we're going to see, all right, that primary oocyte is going to convert into that secondary oocyte, all right, and we're going to get a chance. This is where I told you that one slide, 28.6, or that figure, 28.6, is crucial. If you understand that, read it over a couple times to help you with your understanding of it. All right. So, oogenesis. Before you were even born, okay, while you were developing inside your mommy, all right, our primordial germ cells. These are those diploid cells. All right, those are 46 chromosomes. So at this point, these primordial germ cells, the oogonia, are undergoing mitosis. Okay? They will actually undergo mitosis, all right, and to make more primordial cells. And then from that point, the primordial cells, all right, they've obviously produced their primary oocytes. Then each of those primordial cells will start to undergo meiosis. So we're going to start to develop our oocytes, but they'll stop here in prophase 1. They don't really get a good start. Okay, they just go into the first phase and then they stop. Right, and then you're born, and you spend a lot of your life in prophase 1 up until age 11 or 12. Okay, but important thing is, all right, these primordial cells have the primary oocytes, and this is what that number I was telling you, 1.5 million that you're born with. All right, and so when you enter into puberty, okay, and you start the menstrual cycle, then, all right, we're going to start to engage in the continuation of meiosis all the way up into metaphase 2, okay? When we'll stop until fertilization occurs, and then we'll talk about what happens after that, okay? So when we get to childhood here, all right, again, all right, a little kid, 3-year-old, 4-year-old, 7-year-old, all right, no follicular development is occurring here, okay? But what will happen is that 1.5 million primordial follicles, all right, will regress to roughly about 400,000. okay, which is still plenty. All right, many, many, many. All right, so you go from 1.5 million down to 4,000. All right, this process, this regression is called atrasia. Okay, still a lot, even though 1.5 million is a heck of a lot. All right, you're going to lose, all right, quite a bit. You do the math. All right, over 1 million. All right, then here, when we enter into, at this point, puberty comes. And now we're going to start to see, all right, once puberty arises, now we're going to invoke the endocrine system, okay? So, of course, we're going to deal with the hypothalamus. Our master gland is going to be producing, all right, the gonadotropin releasing hormone, GnRH, all right, which is going to stimulate the follicle stimulating hormone and luteinizing hormone. Now we're going to undergo the ovarian cycle. And there's 3 phases to the ovarian cycle. But I'm not going to go into those tonight because I really want to get into it in depth, but I won't have enough time to do what I want to do. So I'll leave you at that. I know you're anxiously awaiting, all right, the completion of this information. But we'll do that next class, okay? And let's do, I'm going to stop this.